Hello everyone, I Dr. Sharmistha Banerjee welcome you all on the SWAYAM course on genetic engineering and its recent advancement. In the next 30 minutes, I will be discussing what is gene silencing. In the last lecture, we have studied the genetic manipulation of microbes, plants and animals, different techniques used for introducing the foreign gene into the host organism. Today, we will delve into an exciting realm of molecular biology, that is gene silencing. Gene silencing plays a crucial role in controlling gene expression, cellular differentiation and even diseases. Now the question is, what is cellular differentiation? It is a process by which the cell attains a function, and it is a very fundamental step for the organizational structure or hierarchy of the living organism. So, here in this 30 minutes lecture, I will try to cover the mechanism of gene silencing, its significance and its application in research and medicine. Gene silencing is basically a technique that aims to reduce or eliminate the production of a protein from its corresponding gene. From the name itself, silencing means the gene is getting silenced, its function cannot be obtained, or we can say it generally describes the switching off of a gene. Function cannot be obtained or switching off of a gene means that the particular product, gene product, could not be synthesized. Now, what is the product of a gene? It is a protein, and again there are several steps involved for conversion of a gene to a protein or for the expression of a gene into a protein. So, it describes the switching off of a gene by a mechanism other than mutation or other than genetic modification. That is, a gene which would be expressed under normal circumstances gets switched off by the machinery in the cells. When a gene is expressed, we are saying that it is in turned on condition, it is in switched on condition, and here the circumstances is getting just opposite as compared to the expression of the gene. That is, this process will be termed as gene silencing. It occurs when the RNA is unable to make a protein during translation. We all know, and I will show you in the next slide, the quick recap of the central dogma of life. We know that from DNA, RNA is produced and the process is known as transcription. From RNA, the protein is synthesized and that process is known as translation. This is what is central dogma of life. So, the gene silencing process is altering this particular central dogma of life, and again it is useful or it is advantageous in many aspects, but there are certain limitations or challenges of this particular process. So, we will discuss one by one in detail in next slide. Gene silencing is same as gene knockdown, but it is totally different from gene knockout, and this technique we will be discussing in detail in the next lectures. So, I already told you about the central dogma of life. Here the gene expression is directly linked with gene silencing, and it is a very important step in regulating the gene expression for proper cellular function. I already told you how the cellular differentiation is linked with cellular function. Gene silencing, in simple words, we mean that the RNA is capable of not producing a protein, is incapable of producing a protein. That means RNA makes no protein, that is, the gene is silenced. Here is a pictorial representation of the comparison of both the scenarios. In the first case, we have a normal protein production scenario where from gene the mRNA is produced and protein is being formed. But in the second case, the gene is being silenced and there can be different mechanism of it, and because of that the gene is unable to express and no protein is being synthesized, means mRNA is being formed, the gene is transcribed into mRNA, but because of the certain reasons, certain conditions, it can be deliberate as well as it can be natural. So, this is the fundamental steps or key step in the process of gene silencing, we can say. The history of gene silencing dates back to 1990 by Jorgensen. Richard A. Jorgensen, who was born in 1951, is an American molecular geneticist and an early pioneer in the study of post-transcriptional gene silencing. Here it was studied to deepen the pigmentation in petunias. Petunias are basically a very beautiful class of flowers which are purple in color. So, introduction of endogenous gene often resulted in plants with both genes suppressed, like the foreign gene as well as the gene which is already present, both of them will be suppressed, and the phenomena is known as co-suppression. And because of this phenomena, there was the degradation of endogenous as well as the transgene mRNA, means the endogenous gene is also getting degraded when it is being inserted, when a transgene is being inserted. So, this was first time studied in 1990 by Jorgensen. In 1995, Guo and Kempfus, they studied by injecting either the antisense and the sense RNA in germline of C. elegans, and it was equally effective at silencing for the homologous target gene. In 1998, Mello and Fire, they provided the extension of the above experiment, and here both sense and antisense RNA were combined, and it was 10 times more effective than single stranded RNA. Now the question is how the gene silencing works, or what is the mechanism behind the process of gene silencing. Some early findings regarding GS or gene silencing occurred when a team of researchers tried to obtain transgenic petunias with greater amount of anthocyanin pigment. This was the pigment which was responsible for producing color to the flower by amplifying the gene activity of an enzyme, that is basically chalcone synthase. Instead of obtaining deeper purples in the petal, white or chimeric flowers were produced. White or chimeric mean the in between. It consists of both the genes or both the colors were expressed, or white sketchy lines were present at the center of the flower. Apparently, the transgene was not expressed and ended up silencing a homologous endogenous gene. The phenomena named co-suppression was unstably transmitted within generation, leading to the hypothesis that it was mediated by a nucleic acid. So, this was the base for the discovery of the gene silencing, because if it occurred by chance, it may be something which is not carried out from generation to generation, but here in this case the co-suppression phenomena was unstably transmitted within generation. So, it was presumed that there is some sort of nucleic acid which is mediating the process, presumably RNA. Similar phenomena was named quelling in fungi and RNA interference in Caenorhabditis elegans. The research indicated that the presence of double-stranded RNA, a non-occurring form in normal cells. In normal cells, we have only single-stranded RNA or double-stranded DNA. So, this presence of double-stranded RNA, a non-occurring form in normal cells, was related to the silencing of sequence homologue gene. The fact that the phenomena seemed to be triggered by the presence of double-stranded RNA suggested that this could be originally a defense mechanism against viruses and transposomal elements. What do you mean by transposomal elements? They are basically jumping elements or jumping genes, since they originated double-stranded RNA. So, likewise, step by step the principle or the mechanism behind this was discovered. So, most of the gene silencing phenomena are related to RNA activity within the cell. Therefore, the term RNA silencing is often used to describe gene silencing and comprises all the mechanism by which RNA sequences regulate gene expression, except those sequences which are characterized as mRNA, tRNA or ribosomal RNA, that is messenger RNA, transfer RNA or rRNA or ribosomal RNAs. Genetic and biochemical studies have confirmed that the mechanism of RNAi, that is RNA interference, co-suppression and virus induced gene silencing, all of them they are similar. Moreover, the biological pathway underlying double stranded RNA induced gene silencing exists in many of the organisms, if not many, in most eukaryotic organisms. What are the characteristics of gene silencing, or how we can identify the gene has been silenced? Gene silencing is a general term describing epigenetic processes of gene regulation. What is epigenesis? That I will discuss in further slides. This process is important for the differentiation of many different types of cells. Genes are regulated at either the transcriptional level itself or after transcription, or we can say at post transcriptional level. Transcriptional gene silencing is usually the result of histone modification, creating an environment of heterochromatin around a gene that makes it inaccessible to transcriptional machinery, that is the enzymes which are responsible for transcription like RNA polymerase and several transcription factors. Because of this heterochromatin region, or you can say because of the histone modification, the transcription is not possible and gene gets silenced at the transcriptional level itself, or we can say the gene is not being converted into RNA. There are various gene silencing methods currently employed in research and being developed as potential disease therapeutics. Nearly all of them involve disabling the function of mRNA by preventing it from being translated into a protein. However, they differ in the design of molecule used to disrupt mRNA and the manner by which the mRNA is being broken down. As a result, different silencing methods have specific advantages and drawbacks. Based on our need, they are being discovered or used in the genetic modification of an organism. Two of the leading and most understood methods of gene silencing are being categorized as transcriptional gene silencing, that is in short they are abbreviated as TGS. Second one is post transcriptional gene silencing, or they are abbreviated as PTGS. Here is an example which shows how the gene is being silenced, or I am not going into the pathway. Here we can see there is a nucleus, there is a cell, we have a nucleus, till mRNA it is being synthesized in the nucleus, and on the left hand side of the figure we can see that there are certain complex which are getting attached, and this RNA becomes inaccessible for the translation process here, whereas inside the nucleus the DNA, because of the heterochromatin region, it becomes inaccessible for the transcription process. So, these are the two ways by which the gene silencing can happen. Transcriptional gene silencing, they are basically the result of histone modification, already I had discussed. There are different types of gene silencing. Under transcriptional gene silencing, we have genomic imprinting, paramutation, transposon silencing or histone modification, position effect, DNA methylation. And the second category, post transcriptional gene silencing, includes RNA interference, RNA silencing, nonsense mediated decay. So, today I am going to discuss the different types of transcriptional gene silencing, whereas in the next lecture we will focus on the post transcriptional gene silencing mechanisms or methods. Genomic imprinting, the first one, is an inheritance process which is independent of the classical Mendelian inheritance. It is an epigenetic process that involves DNA methylation and histone methylation without altering the genetic sequences. Histone methylation, histone are basically the proteins which are responsible for condensing or making the DNA stable. Now, the question is, what is epigenetic process? Epigenesis is a process by which plant, animals and fungi, they develop from a seed, spore or egg through a sequence of steps in which the cells gets differentiated and organs are being formed. This process is in contrast to preformationism. It is basically preformationism or preformism is basically a popular theory that organisms developed from miniature versions of themselves. In this case, that is, they do not develop from egg, sperm or a cell. So, this is what is epigenesis is. It has been demonstrated in epigenesis has been demonstrated in insects, mammals and flowering plants. So, likewise the gene silencing can also be observed in these individuals or organisms, we can say. Genomic imprinting is basically a process by which only one copy of a gene in an individual, either from their mother or father, is expressed while the other copy is suppressed. So, unlike genomic mutation that can affect the ability of inherited genes to be expressed, genomic imprinting does not affect the DNA sequence itself. Instead, gene expression is silenced by the epigenetic addition of chemical tags to the DNA during egg or sperm formation. Epigenetic tags on imprinted genes usually stay in place for the lifetime of the individual. Here is a figure wherein we can see the example of genomic imprinting. In the purple color chromosome, it indicates the maternal chromosome, whereas the blue color chromosome, it indicates the paternal chromosome. So, here we can see in the left half the maternal chromosome is being imprinted, whereas in the right half the right half of the figure, the paternal chromosome is being imprinted. So, this is an example of genome imprinting. Only the one half of the chromosome or the genetic material is being expressed, the other is being suppressed. So, this is again a form of gene silencing and it is at the transcriptional level itself. Paramutation. In epigenetics, paramutation is an interaction between two alleles at a single locus, whereby one allele induces a heritable change in the other allele. The change may be in the pattern of again DNA methylation or histone modifications. It was first observed by the effect it had on the color of the corn kernels in maize plant. Then the next type is position effect. Paramutation I will discuss in detail in the next slide with the help of a figure. Position effect, it is the effect on the expression of a gene when its location in a chromosome is changed, and it is often by translocation. This has been well described in Drosophila with respect to eye color and is also known as position effect variegation, that is abbreviated as PEV. Here this is an example of transposon gene silencing as well as paramutation. So, what we can see that there are two types of chromosomes which are involved here, chromosome A and chromosome B. In the first case, the transcription begins, the chromosome A is being transcribed in both the cases. Then the chromosome A is being indicated with red color as well as the chromosome B is indicated with blue color. So, what is happening in the first half, that is the gene pattern is being expressed. How? Because the chromosome B is being converted into the DNA and next in the RNA with the help of reverse transcriptase enzyme, and it is being incorporated into the open reading frame. Whereas in case of second case, that is in case of paramutation, the chromosome B is not reverse transcribed, it is not incorporating into the open reading frame, whereas it is blocking the open reading frame, and as a result the gene is getting silenced. RNA directed DNA methylation is prevalent in flowering plants and induces transcriptional silencing in repetitive DNA, which includes all types of transposons. Transposon silencing is a form of transcriptional gene silencing targeting transposons. Transcriptional gene silencing is a product of histone modification that prevents the transcription of a particular area or particular segment of DNA. Transcriptional silencing of transposomes is very crucial in maintaining a genome. Jumping of transposons generate the genomic instability and cause the extremely deleterious mutation. Transposomal elements insertion has been linked to many diseases including hemophilia, SCID, severe combined immunodeficiency and also predisposition to cancer. Here is an example of the transposon silencing. Here is a figure that represents transposon silencing. We have a double stranded RNA, then we have dicer which is acting on it, and in the other case we have DRM, that is basically methylating this particular RNA, or we can say they are a class of methylases which are modifying this double stranded RNA, or DRM is basically DNA and RNA dependent methylases. So, in the left hand side we can see we have dicer. So, it is cleaving the double stranded RNA into small fragments. Then this dicer in present this diced mRNA, from the name itself dicing. So, this diced mRNA, or we can say siRNA, miRNA, RNAi, all these incorporates with RISC, that is RNA induced splicing complex, and then they further creates splicing in the segment, and in this way the gene silencing is achieved or gene silencing occurs. Transgene silencing, unfortunate insertion of transgene into a transcriptionally inactive part of a genome. When an insertion of any transgene, it does not show activity as per the desired result, this is because of its instability. The loss of transgene stability is again because of the gene silencing phenomena. Example, slow fruit softening tomato was obtained by reducing the expression of the ripening enzyme, that is polygalacturonase enzyme, and it is again a beautiful product of gene silencing or transgene silencing, we can say. Post transcriptional gene silencing, they are also known as PTGS or RNA interference. It is a mechanism of regulating gene expression in wide range of eukaryotes. Herein, small RNAs block the gene expression by targeting homologous mRNAs without impacting the nuclear DNA. So, this I will discuss in detail in the next lecture. Advantages of gene silencing: down regulation of gene expression simplifies the knockout analysis, easier than use of antisense oligonucleotides, that is siRNA, more effective and sensitive even at lower concentrations. It is a cost effective phenomena. High specificity middle region, that is 9 to 14 are more sensitive with siRNA. The researcher can simultaneously perform experiments in any cell type of interest. They can be labeled. Ease of transfection can be achieved by use of vector blocking the expression of unwanted genes and undesirable substances. Inducing viral resistance, powerful tool for analyzing unknown genes in sequence genome, useful approach in future gene therapy. Oligonucleotides can be manufactured quickly, some within even 1 week. The sequence of the mRNA is all that is needed. So, these are the very important advantages or applications of gene silencing, we can say. Disadvantages of gene silencing: the first one is high pressure injection and electroporation can cause significant injection damage to the integrity of the normal tissues and organs, and thus preclude the utilization in a clinical step. Thus, they are not preferred for clinical studies. Liposomes or cationic encapsulated small interfering RNA may also be toxic to the host and may cause severe host immune responses. Other emerging strategies include chemical modification of siRNA molecules and encapsulated with different molecules that are still in their infancy and need to be thoroughly investigated before they can. Let's have a quick discussion about dicer and RISC. Why these two? Because these are the important tools for RNA silencing or gene silencing. Dicer is basically RNase 3, that is, it is an enzyme which is acting or digesting the RNA from the name itself, RNase, like double stranded RNA specific ribonuclease, enzyme involved in the initiation of RNAi or RNA interference. It is able to digest double stranded RNA into uniformly sized small RNAs like siRNA or even heterogeneous RNAs or hnRNAs. Dicer family proteins are ATP dependent nucleases. RNase 3 enzyme acts as a dimer. Loss of dicer means loss of silencing, which is being processed in vitro. Dicer homologues exist in many organisms including C. elegans, Drosophila, yeast and even humans, where dicer is actually a conserved protein. Dicer is a ribonuclease belonging to RNase 3 family with 4 distinct domains. The first one is amino terminal helicase domain. Second one is dual RNase 3 motifs in the carboxy terminal segment. The third one is double stranded RNA binding domain, and the last domain is PAS domain or PAZ domain, which comprises of 110 to 130 amino acid present in protein like ergo PV. Basically, it is thought to be important in protein-protein interaction. Without this domain, dicer cannot bind to the RNA and it cannot interact with the other RNA interfering proteins or enzymes. So, it cannot act if it lacks PAS domain. It cannot cleave the RNA if the PAS domain is absent. The second important tool for gene silencing is RISC. That is, I already told you, it is RNA inducing silencing complex. RISC is a large, that is having a molecular weight of around 500 kilodalton. It is a large RNA multi-protein complex which triggers mRNA degradation in response to siRNA. Unwinding of this double stranded siRNA by ATP independent helicase occurs here. The active components of RISC are endonucleases, which are also called as argonoid protein, which cleaves the target mRNA strand. Herein I will discuss a case study why gene silencing was focused, or it is an important tool for therapeutic strategies. So, morpholinose are one of the tools scientists have a lot of experience with when it comes to modifying the zebrafish DNA functions. With a fully sequenced genome and thousands of genetically modified examples already available, these small fish are a prime example of using morpholinose for gene silencing in drug development. The use of zebrafish in early drug discovery and in particular preclinical studies has seen a significant amount of modified zebrafish DNA through gene silencing with morpholinose. In the next slide, I will discuss what are morpholinose. Through the controlled use of morpholinose, it is possible to see exactly how a trait will manifest itself in the biological system. Morpholinose are basically oligomeric molecules that are useful for modifying the gene expression. These specialized molecules give researchers and scientists the means to block the excess of specific RNA and then silence the targeted genes for precise experimentation. So, using morpholinose to silence this specific gene makes it possible to learn about very specific interactions between the DNA and gene expression, between the DNA as well as the translation process, we can say. Once a gene is silenced, the other traits of DNA and how a new drug or disease affect it are easier to observe. Morpholinose allow for viable research into gene expression, connecting specific traits to specific genes. Sharing homology with humans means that the results of experiments, testing and arrays with these modified zebrafish are highly applicable to real world diseases that impact humans. Ensuring viable data with repeatable experiments on specific genes help create a full understanding of how a new drug could affect humans. It also helps in finding the best way to use gene editing and gene silencing to help the drug discovery, and it is essential to stay ahead of the fast moving pharmaceutical industries. Having the right animal model ready for any assays allows for a smoother transition into the later stages of drug development. In practical terms, that translates into zebrafish, allowing for more new treatments to enter the market quicker. In the next lecture, we will study in detail about the post-transcriptional gene silencing methods that involves RNAi, miRNA and siRNA, as well as many more mechanisms involved in it, and the examples by which the gene silencing helps in creating different transgenic plants and animals. So, thank you for joining us in the enlightening journey of gene silencing.